So we're moving on to our next presenter now, Glyscend Therapeutics. We've got Ashish Nimgaonkar, MD, President and CEO. Ashish, I'm very sorry if I've messed up your surname, but perhaps you could pronounce it properly for me.
Sure. First of all, can you hear me okay?
Yes.
Great. Ashish Nimgaonkar.
I was nearly there.
Very close. Can you guys see my slides here?
Yeah, we can see your slides. We can't see you on full screen. If you just maybe swap displays now, click that swap displays button, that'll be ready to roll, I'd imagine.
Phil,
yeah, there we go.
Phil, as Ashish gets his slides up, let me just tell everybody that if they are on Cadence, they can go to the poster session and they will be able to see his slides and follow his slides. Even on Cadence, that might even be clearer. And that's the case for everybody coming after him now. So if you're interested in following this up on the poster session, take a look at it right now, and you'll also see some of the posters too. But it might be easier even to follow your presentation. So go right ahead.
Great, thanks a lot for giving me this opportunity. So Glyscend is a clinical stage venture-backed therapeutics company where we are advancing first-in-class polymer-based therapies targeting the gastrointestinal mucus as a target. And can you see this slide?
Yeah, we've got it.
Yeah. So this concept and technology really bore out of some of my work in collaboration with folks from Geltex and Hopkins. I'm a gastroenterologist, scientist, physician, still practicing medicine. But many years back, we were working on a problem of targeting metabolic diseases with a novel concept. And we started working on this concept with deep insights from folks who have worked at Geltex, which was one of the blockbuster biotech companies in the 1990s, which was a pioneer in developing the first GI targeted polymer therapies for hyperphosphatemia and hypercholesterolemia, Renagel and Velcol, which continue to be a significant market success even today. And so what over the past many years, what we have really developed is a platform where we are able to synthesize novel polymers, fine-tune it for targeting various segments of the gastrointestinal tract, where these polymers have the innate ability to complex with mucus, which is the natural barrier, as you may know. And now what we have is a polymer platform where we are advancing in two distinct asset classes. The first is polymers as therapies. And the first candidate is in clinic as we speak in phase 1 trials for targeting type 2 diabetes and obesity. And we have other candidates in the pipeline where we are using them for targeted drug delivery, both for indications within the GI tract as well as for targeted peptide delivery. And the fundamental concept is as follows. The polymers that we have developed, they complex with mucus, unlike other technologies which are essentially mucoadhesives, which are surface binding polymers. Our polymers actually integrate with mucus and enhance the natural barrier properties of the mucus. So on the right-hand side, you'll see that after we mix our polymer with the mucus, it creates a thick gel which stays within the GI tract. We did not intend to develop this technology platform. The real goal here many years back was really to understand and develop a solution to target mechanisms of bariatric surgery. And as a gastroenterologist, when I looked at this problem of how do we tackle the metabolic syndrome at its root, the key was in the GI tract. And the learnings here were really from Roux-en-Y gastric bypass surgery, where we found that patients with not just obesity, but type 2 diabetes who underwent Roux-en-Y gastric bypass surgery, they were off their insulin and many of the anti-diabetic medications within few days after the surgery without having lost any weight. And also over a long period of time, this surgical approach has had durable response in patients with type 2 diabetes. And the key part of the gut, which is instrumental, is the first portion of the small intestine, which is called the duodenum. And furthermore, in the last decade or so, there have been this mechanism of duodenal exclusion has been translated into a number of therapeutic opportunities, particularly the endoscopic space, where there are sleeves which are endoscopically placed in the duodenum, and also there is ablation devices. And all of these technologies have actually proven out the human concept of inducing duodenal exclusion in patients with type 2 diabetes, where they have shown robust reduction in postprandial glycemia as well as long-term hemoglobin A1C reduction. So when we are looking at this problem and realizing that there are limitations to both bariatric surgery as well as endoscopic devices, we thought that why not we target this mechanism with a pill? And therefore, we came up with this non-invasive way to target this mechanism of duodenal exclusion. And this is how it works. A patient takes a pill preprandially. The pill goes into the gastric cavity. It dissolves the polymer. It's a solution form, but as soon as it goes into the duodenum, there is a pH shift. And the polymer has a phase transition, integrates with the mucus, complexes with the mucus, and creates this quote unquote barrier, and essentially enhancing the natural barrier properties of the duodenum and the proximal jejunum. And the key property of the polymer is it's completely inert. It's not metabolically broken down in the GI tract, and it transits throughout the GI tract within 12 to 24 hours, and it's not systemically absorbed. In the lower half of the slide it shows the phase transition at different pHs, where around pH of 6 to 7, which is the pH of the duodenum, this polymer becomes insoluble complex and changes the properties of the proximal small intestine. This is how it looks once you administer it inside the GI tract, and this is a cross section on the left-hand side, where we have shown with fluorescently tagged polymer that it creates a circumferential coating or a barrier in the duodenum, and which stays for many hours after administration. We have demonstrated the efficacy of our lead candidate in numerous preclinical models of type 2 diabetes and obesity, which are very well validated for this mechanism of duodenal exclusion. Moreover, we have also completed a long-term 28-day GLP tox in 2 animal models, and we have advanced that candidate into the clinic in the ongoing phase 1 clinical trial. The right-hand bottom graph basically shows that this candidate is completely eliminated in the feces without any systemic absorption. And so while we are advancing our lead candidate into the clinic, we will be initiating a patient study early next year. We are also advancing other candidates for other metabolic indications, as well as working on programs for using the polymers as drug delivery vehicles to target both indications within the GI tract, but also for oral delivery of peptides. And so in summary, what we have developed is a novel first-in-class mucus complexing polymer technology, which we are now advancing in 2 distinct classes, both as polymers for primarily treating GI and metabolic disorders, but also polymers as drug delivery vehicles. With that, I'm open to any questions.
That's wonderful, thank you. So there's a lot of work that you're obviously really putting in there, Ashish. So in terms of next steps for the organization, do you have sufficient funding now to take you on to the next stage of your R&D pipeline as well as your growth?
Yeah, we are fortunate to have a very strong group of syndicate investors, but money is never enough. So we are initiating our next round of Series B financing pretty soon.
Okay, great. And what sort of level will that be at, Ashish?
It's in the $50 to $75 million range.
Right, got it. Question came in, says, does it work with high cholesterol? Is a question.
High cholesterol?
Probably, it looks like cholesterol to me, but cholesterol is what the person's put. I'm sorry.
Well, given the underlying mechanism, it should, but it needs to be proven out in our clinical trials.
Yeah, good. Okay, well, thanks so much for your time today, Ashish. No more questions. Oh, one more question's coming from Gordon Cutler, which is, does the Glyscend product have similar decreases in protein, fat, and lipid absorption?
We haven't tested that, but that's a good question. Something that will play out again in our clinical evaluation.
Great. Well, best of luck with the next stage in the clinical pipeline and the fundraise that's coming up. So thanks very much for your time today, Ashish.
Thank you.
Great. Thanks.